Welcome to this movie of HIP Building Block 3. This is the third building block of the Holland Innovative Potato project. Within this building block we do research on disease resistance in potato. Well, there are many diseases that are troublesome for potato production. There's, for instance, the late blight, which is a very notorious one. But there's also nematode, there's black leg disease, there's fusarium in tubers, aphids, insects, and potato Colorado potato beetle. So there's many diseases in potato cultivation that need to be controlled. So for some of the diseases like late blight and for insects, there is control, but that requires a lot of pesticides to be sprayed in the field. And of course, if we can prevent that, it is more preferred. So we would like to have, in fact, potato plants which are resistant to these pests and diseases. And now, how can we make those potato plants? Therefore, we need to find natural resistance genes. Those genes can then be introduced into potato varieties through a breeding process. But for such a breeding process, of course, you will need to have sources for making the crosses. So genetic resources for the resistance. And for some of the diseases, this is already quite well studied, like for late blight, there are quite some sources available for nematodes. Although with these diseases, there's already currently there are problems with strains and isolates that can overcome the diseases. So the new virulent nematodes that are emerging currently face new problems for breeding. So, but there's also many diseases like black leg and insect resistance, insects for which no resistance is available yet. And exactly for those diseases, we would like to do new screens for resistant sources. And that is what, in fact, this building block is about, to screen wild relatives of potato for those resistances to less common diseases and to diseases for which currently no resistance is available. Wild relatives of potato, they mainly come from South America, Central America, because the wild relatives from potato in Europe, they are not so suitable. And there are many seed banks or gene banks available which harbor those Solanum species, and one sub-collection of this Solanum collection is available in plant breeding. Currently we have 500 of those accessions, and those accessions we will screen in the HIP project. And yeah, for doing those screens, we need to screen so many plants, we need very clever screening techniques, high throughput ways to screen for those resistances. And you can imagine that it's not a very easy thing to do in wild Solanum species, which very often don't look like potato so much, and they don't produce tubers under the current under the long day length in Europe. So what we do, we devise clever tricks to screen those Solanum plants. Like for instance for Erwinia, we do not screen the tubers, but we screen for resistance in the leaves. And this experiment that we have here, we are doing such a screening. Here you see a set of plants which have been inoculated 1 week ago in the leaf with Erwinia bacteria, and here is the susceptible variety Ivory Russet, and you can see that the leaves have died. Also here the susceptible Solanum chacovensis species is completely susceptible, as visible by the dying leaves. But here is an accession. This has been treated in the leaves and which shows only minor symptoms, only few brown spots are occurring, and even there are accessions which do not show symptoms at all. So what we will do now is to punch out a hole in the leaf to determine how many of the bacteria can actually survive.
Within the HIP program, we are working on resistance against insects in potato. And this is of course because there are problems associated with the use of insecticides in the field. And we try to develop new approaches to circumvent that. And one of these approaches is host plant resistance against insects. So we work on resistance against the aphids, whiteflies, and the Colorado potato beetle. Yeah, as you all know, the potato is not resistant against any of these insects, so we need to find new resistant sources. And these we find in the wild relatives of the cultivated potato. The wild relatives come from South America, and a lot of them have resistances in them. And what we do here now is first screening all these wild relatives for resistance against these 3 insects. And today I will show something about the resistance screens that we do against the Colorado potato beetle. And here you see the plants that we use for this assay. These are covered in a sleeve because we want to put the Colorado potato beetle on the plant, and the beetle has to feed on the plant. So it cannot feed somewhere else, it must feed on the plant. And if it cannot feed, then it dies. And then we consider the plant being resistant. Of course, for this you need the beetles. And we are rearing these beetles here in Wageningen. And from the beetles we actually do not use the adults. We use the larvae because we think that using the larvae is the best way to find out where resistance is present, because the larvae are the most vulnerable stage of in the life cycle of a Colorado potato beetle. So what we have in our rearing is adult beetles. They lay eggs, and in nice packages we collect these eggs, and after 5 days these eggs hatch, and the 1-day-old larvae are put on the plant, and they are allowed to feed for about 9 to 10 days. And that's just to about pupation. And after these 9 days we first determine whether the larvae are alive or dead, and if they are alive we weigh them. So the weight of the larvae is a measure for the resistance, because if they feed not, then they die. If they feed poorly, they usually have a strongly reduced weight, and that is the measure that we use for finding the resistance. So we screened more than 300 different accessions for resistance against the Colorado potato beetle, but also against the aphid and the whiteflies. We found a resistance against all of them. And in some cases we found actually plants that were resistant against all 3 insects. So these are really, really interesting. The next stage in this research is, of course, that we try to identify where the genes are in the genome of the wild potato, and then we will find molecular markers linked to these genes, and the breeders can then use the plant together with the markers to introduce the resistance gene in the cultivated potato. And then hopefully they will bring new varieties to the market that are resistant and do not need any insecticides anymore.
We are here in Nematology lab, and here we do a resistance testing against nematodes. In this HIP project, we are mainly interested in the root knot nematode, Melodogyne chitwoodi, and the potato cyst nematode, Globodera pallida. Mainly what we do here is a bioassay, an in vitro test, and after the initial screening here and getting the results, we will go on into the lab in the POTI experiment, and then ultimately, after confirming the resistance in the POTI test, we will go into the field test. So normally, if you see closely, this one is a gelatinous matrix. It's a fluid-like. You can already see it with your naked eye. And this female already laid like hundreds of eggs already in this gelatinous matrix. If you see also the opposite, this is again the apple-shaped female of Melodogyne chitwoodi, smacked MC31. And this is the egg mass, which is light. So this is also one of the susceptible accessions that we have. And this is the feeding cell, which is so-called giant cell. You can see that already it is bulged out and it forms a big gall. Here we can already see some of the results we have for the first phase of the experiment of nematodes in the HIP. In total, we have tested about 129 accessions, 6 replications. In total, with the 2 nematodes that we have, this contains about 1,600 plantlets of potato, which are already tested in the first phase. And we have already the results. And quite nice accessions are there which are resistant to both Melodyne chitwoodi and potato cyst nematodes, Globodera pallida.
Yeah, so the HIP project is now just in its startup phase. We have already identified several accessions with resistance to Erwinia, to Colorado potato beetle and virulent nematodes. But that's still the beginning. Now we have identified those resistant accessions. We will try to find out which of them are most suitable for the breeding practice. So how many genes are involved, where are the genes in the genome, and how easy do those resistances transfer to next generations in the breeding process. But that's quite some work to be done still, but fortunately the HIP project is still running for several years. Also a very good situation is that this is a public-private partnership with commercial breeders involved, and they will in the end use these very good resistant accessions for their own breeding programs, and then they will select varieties which are suitable for commercial cultivation.